Uitgenodigd vanuit Wageningen, Melissah Rowe en Ruth Fawthrop. Ze zullen ons meenemen in het verhaal over de Italiaanse mus en de huismus. Ik vind dit echt typisch zo'n soort waar je toch een beetje als twitcher zijnde aan je achter je oren moet gaan krabben van wat ben ik nou aan het doen. Want elke huismus kijken we eigenlijk niet naar om. Maar zodra het Italiaanse neefje is, dan is het ineens wel echt een heel gaaf beest. Ik bedoel,
voorzichtig zijn.
Kijk even. Fantastisch. In ieder geval wist Melissah me aan de telefoon te garanderen dat ook huismussen echt hele leuke vogels zijn. Best wel brutale beestjes, een beetje de mafioso onder de vogels. Dus wat dat betreft is die hele Italian job wel een mooie naam voor het onderwerp. Ik durf wel te zeggen dat de volgende keer een huismus weer je appeltaart aan het opvreten is op een terras. Je gegarandeerd wel even terugdenkt aan dit verhaal. Ladies and gentlemen, the Italian Job Explained by Melissah and Ruth.
Thank you very much. I think everyone can hear me okay? Yes. Great. Okay. So first, I really want to thank everybody for having me here today. It's a real pleasure and a real honour for me to be able to speak with you. I really do enjoy when my science can interact and mesh with people who are out looking at birds in the field, because I think those two things are really, really complementary. So my name is Melissah. I am from the Netherlands Institute of Ecology. I think you can tell I'm not Dutch though, so I apologise for I can't speak Dutch, and now I can't even remember how to say I can't speak Dutch because I'm nervous. En beetje Nederlands. So I wanted to talk to you today about this funny bird, which I think is a really, really cool bird. But I want to start off, first of all, just by telling you that this is not just my work, that there's a group of people that are involved in this story I'm about to tell you, and all their input has been just as important, if not more important, than my own, including the people that helped me actually go to this site and work with this individual bird. And the photos that you see of this bird are also not my own. So, why am I here? I'm going to tell you this story about this strange little bird. This guy popped up in a garden in Garderen in 2022, and everybody got really quite excited because it seemed to be the first instance of an Italian sparrow here in the Netherlands. So he seemed a little bit far from home, but vagrant birds do indeed pop up in different places. And for something like a little seed-eating sparrow, a truck or a boat that has some animal food on it seems like a pretty nice place to be. So it's not impossible that a little bird could hitch a ride from Italy and end up in the Netherlands. But you see, Italian sparrows, they're kind of a really interesting little bird. And so for someone like me that studies sparrows, what I know about these Italian sparrows makes an Italian sparrow turning up in the Netherlands even more interesting than I think just this is a really exciting vagrant bird. So why is that? So probably you guys know the story, but let me give you a little background. The Italian sparrow is a bit of a special species. It's a species of sparrow that's found in mainland Italy, but also on the Mediterranean islands. And it's special because it is one of the few cases of what we call homoploid hybrid speciation. So what does that mean in plain English? It basically means it's a hybrid species. It means that the Italian sparrow has arisen as a result of hybridization between the good old house sparrow and the Spanish sparrow. One of the things that's really nice about the Italian sparrow, and one of the things that actually led to the original hypothesis that it was a hybrid species, is it has a very intermediate phenotype, which means it shares plumage characteristics with both of its parental species. So it's a really good blend of the two. And it's actually, so it's this intermediate phenotype which led people to think that it was maybe a hybrid species. And some work by my collaborators subsequently has shown at the genetic level that this is indeed the case. So let me tell you a little bit about that and how they know that. So what they did is they looked at the genomes of these 3 different birds, house, Spanish and Italian sparrow. I'm going to show you a couple of graphs here, and I just want to walk you through how to read them so that they make sense. But if anyone needs any clarification at the end, I'm happy to take some questions. So basically what this plot does is when you look at an individual dot, this is an individual bird, and that dot represents the entire genome of that bird. When dots are closer together, they're clustered together, it means their genomes are more similar to one another. So what we see on this plot is you see a cluster up here, which are house sparrow, you see a cluster over here, which is Spanish sparrow, and really nicely, intermediate to that is a cluster of birds, which are the Italian sparrow. So what we can see here is that we don't just have an intermediate phenotype, we have an intermediate genotype as well. So another way to look at this is these sorts of plots. Also another really weird plot, but I hope I can explain it for you. This plot is essentially showing you ancestry. So if you look at all the genes, house sparrow genes are represented in blue. And so if we look at house sparrows, they have all house sparrow genes. If we look at Spanish sparrows, they have Spanish sparrow genes. But when we look at the Italian sparrow, again, we see this real mix of house and Spanish sparrow genes. So what has happened is the Italian sparrow is this nice sort of mosaic or mixing of house and Spanish sparrow genomes. And it's now become a stable species, so it is isolated from both of its parents, so it's a solid, stable species that continues to, it doesn't just collapse, it continues to maintain a population in Italy. So that's the basic story, but it gets a little bit more complicated. And that's because some work that another colleague of mine has done has actually looked at the different populations of Italian sparrow across the different Mediterranean islands. So what she did is she went out and sampled across the islands of Crete, Malta, Sicily, and Corsica. She also compared this to Spanish sparrows and to house sparrows. And what she finds is that as you go across these different islands, the genomes are still this mix of house and Spanish sparrow, but the ratio of that mix varies. So let me walk you through this. Again, if you look at a house sparrow population, that's represented by a blue genome, and a Spanish is represented by a red genome. The example I showed you in the previous slides showed you about 60% and 40% mix of the 2 genomes formed the Italian sparrow. But as we move from Crete, what we see is only about 20%, 30% of the genome is made up of Spanish sparrow genes, and the rest is from the house. And that's a very different story to when we get over to Malta. So you can imagine you take 2 genomes and you mix them together. You can do that in different sort of proportions. Some things, there's some elements that are really important and always have to be the same. But essentially it means there's more than one way to make an Italian sparrow. But the story even gets a little bit more complicated, and that is because if we look, we know the Italian sparrow is this past hybridization event where they've come together, they've hybridized and formed the Italian sparrow. But there's really good evidence that the house and the Spanish sparrow are also still experiencing some level of gene flow. So some genes from the one species go into the other species. So just to give you a little bit of a clue into that, some work that Mark Ravenet has done, and he is working with me on this bird as well, showed that the house and Spanish sparrow, they did diverge from one another a bit less than 1 million years ago. And then we have a really interesting secondary step where the house sparrow that we all know and love, Passer domesticus, diverged from another really interesting bird called Passer bactrianus. You're going to hear a little bit more about this other bird in a moment with, from a talk from my colleague Ruth. So then there was this split over here from bactrianus and house sparrow, but the really interesting thing is the molecular evidence, so looking at the genes of different populations of these 2 species, really does show that there's ongoing gene flow between house and Spanish sparrows. Moreover, it shows that it's asymmetrical. So there's more Spanish sparrow genes coming into the house sparrow populations than the other way around. So there is both reciprocal integration, but it is more biased from the Spanish to the house sparrow. I'm going to show you another one of these plots, and I really just want you to have a take-home message. We've said blue is house, red is Spanish, and that's the case here. But when we introduce this Bactrianus sparrow, we can see things a little bit more from a wider perspective. And what we see is this Bactrianus sparrow really is all house. We have Spanish sparrow over here with just a few house sparrow genes making it into the population. We have the Italian, which is this nice mix between the two. And we have modern-day house sparrows, which seem to harbour somewhere about 10 to 15% of Spanish sparrow genes. So that brings me back to our little friend and the question of, well, what are you? Are you a vagrant Italian sparrow that maybe hitched a ride on a truck? That's a really good hypothesis. But as a scientist and as someone who wants to question things, we have to think other explanations. And so when we first heard about this bird, the first thing that popped into our head is, well, maybe it's actually just a new hybrid, a first generation or F1 hybrid between a Spanish and a house sparrow. We've actually produced these F1 hybrids in captive breeding programs, and they look a lot like Italian sparrows. So looking at this bird, the obvious thing is it's an Italian sparrow, could also be an F1 hybrid, but we were thinking about it and we came up with another hypothesis, which even seemed a little far-fetched to us at the time, which was if you take a handful of Spanish sparrow genes and throw it into a house sparrow genome, there's a lot of house sparrows out there. Maybe every now and again you hit on the right combination of genes that you might have some of the phenotypic effects of an Italian sparrow come through. So we termed it an emergent Italian phenotype, or in other words, a weird house sparrow with just the right combination of genes to look a bit different. I want to point out that this sort of idea has actually popped up before. In fact, in the UK, there was a bird, this little guy here, another special little sparrow, who turned up in the UK and looked like an Italian sparrow. And people have similar thoughts to me. Okay, what is it? Let's take a look. So they caught this bird, it's this guy here. They took some DNA, they took a feather sample. So they had some DNA and they could look at about 6 different genes in this sparrow. And what they found by looking at these genes is that he was indeed a house sparrow. So what this table shows you is here's the different genes that they looked at, and here's the different hypotheses, right? If he was a house sparrow, these are the genes he would have. If he was a Spanish, this is what he'd have. Italian, this is what he'd have, and so on. And this is what this bird had. All house sparrow genes, mitochondria and nuclear genes. And when they then used those data and sort of looked to see how they clustered, this bird also clustered very strongly with all the other house sparrows. So what the genes were telling us was that it was a house sparrow. So we thought, maybe our bird could be the same. So we went back to our bird, asking these questions, and to sort of see whether we could answer it, we decided that we would try and get a sample from him. So with the help of people here and people who are not here, I got permission to go out and work with this bird. We were able to trap him. I must say, having trapped a lot of sparrows in general are pretty tough to trap. So I felt a little disheartened trapping that sparrow, but we were really lucky and we managed to get him in our net and we managed to get a sample from him. And so this little bird had been running around Garderin. He'd also been copulating with what looks like a female house sparrow. Females are really tricky to tell apart, so I assume it's a female house sparrow. But we managed to get him in our net. It was really exciting to have him in my hand. We took a bunch of standard morphometrics. We put a little ring on him. We had a bit of a look. He was in my hand. I was like, looks like an Italian sparrow to me. But the proof is in the DNA. And what the DNA of this bird told us, well, my clusters don't show up, so I'm going to ask you to believe me when I say that in this circle here is a whole bunch of dots that represent genomes from Italian sparrows. We took 2 different populations of Italian sparrows, their entire genomes. We took 2 populations of Spanish sparrows. We took it's a real shame this doesn't show up. We took a whole bunch of samples from house sparrows, both in the Netherlands and in France, as well as some birds that were trapped in the same net as our target bird. We sequenced their entire genomes, we plotted them out, and what we saw was all our Italians were here, so all the dots were really close together. All our Spanish were over here. House sparrows, we saw a little bit of variation that went from different populations, but our target individual is slap bang in the middle of all the other Dutch house sparrows. So, looking at his entire genome, 100% certain that he's a house sparrow. So, a bit disappointing to some, I understand, but also really, really exciting, because then he offers us a little bit of an opportunity to understand more. So what does he offer as an opportunity? He offers us an opportunity to try and understand, well, if he's got the right set of genes to look like an Italian sparrow, what genes are they that makes him look different? Obviously, this is a nice normal house sparrow, whereas this is our friend here. So by having this individual, we can potentially start to look at what regions of his genome are different. So what the analysis we're doing right now is to try and see if we can pinpoint the genes that make this bird different. The way we do that is with molecular data again, and we scan along the genome. I just want you to get a bit of a snapshot of this, not to really understand it. But what we do is we look along the genome and we look for points that spike up. And those spike points tell us at that point in the genome, this bird is more Spanish-like than it is house-like. And once we have those points for this bird, we then see if that is the same points in any other Dutch bird. And where the normal Dutch birds and this bird differ is where we're going to dig deeper in and look and see what genes are there. And our hope is that maybe we can begin to understand how the genotype relates to the phenotype. And what's really cool with this sparrow group, the 3 species, because they've got a genome mix in the Italian sparrow, and there's integration between the house and the Spanish, what's really cool is we've got this whole sort of system where we've got variation in the genetic material, and we can start to relate that to things like their plumage, but also to their behaviour. These birds do act a little bit differently, to things like morphology, their basic ecology, whether they migrate or not. So sparrows are a really fun system to work with because you have this really amazing capacity to try and link what the genome says and what the bird looks like. But I think sparrows are really cool for a number of other reasons, and my colleague Ruth is going to tell you a little bit about them, but just to give you a little bit of a highlight. House sparrows, I think we all know, and I hope we all love, I certainly do. They're a really common part of our life. We see them at cafes, we see them at zoos, we see them stealing our bread. So they've got this really long association with humans that makes them really interesting. And that's what Ruth is going to tell you a bit about. But there's also this really interesting case of when is a house sparrow not a house sparrow. And other than our special bird here, there is another really interesting case, which was this bird here that I alluded to. And so that's this bird here, which is also a house sparrow, but it's a different subspecies of house sparrow. It's called the Italian sparrow. And so we were recently in Kazakhstan working with this bird, and Ruth's work really delves into this. But we have this really interesting system where we've got a house sparrow that also doesn't act a whole lot like a house sparrow. So there's a lot of really good and interesting questions that we can ask. So I kind of wanted to finish up going back to these vagrant Italian sparrows, and just to say to you, we want your Italian sparrows, your vagrant Italian sparrows. And we've already started to get some, which is really exciting. So first of all, we've reached out to the people that worked on this UK bird to see if they have some more DNA that we could look at the whole genome on. I've also recently had someone contact me from Argentina saying, we have an Italian sparrow, but I read your thing and maybe it's not an Italian sparrow. So they're going to try and trap it and send us some blood. And then also we've been working with some people in Germany, don't have a picture of the bird unfortunately, who have 3 different cases of putative Italian vagrants, and they've also sent us some samples. And what our hope is that we can get enough of these birds, we might really be able to say something interesting about the genes for, say, the crown coloration. So this is my call to you. If you are somewhere in the world and you see an Italian sparrow and you think, maybe you're a little far from home, give me a call. And with that, thank you very much.
Thank you so much. Before we move on to the next part of the story, I was wondering if there are any questions. Questions can be asked in English or Dutch as well, and we get a translation. So feel free.
Thank you for this very interesting talk. I'm a bit puzzled though, because I still don't understand if we talk about the Italian sparrow, if its phenotype is clearly showing we are looking at a species, you could say, a field birder, but the genotype doesn't tell us it's a hybrid. So where is this, when do you talk about species in this sense?
Oh, that's such a huge question. Let me first of all say that with the Italian sparrow, you do have this phenotype, right? And you have a genome which is a mixture of 2, but it's a stable genome, right? So there's a tiny bit of interbreeding back with, say, house sparrows in the Italian Alps. There's a little bit of hybridization. We do see the odd little bit of hybridization in other places. Spanish sparrows have just started to move into the Gargano Peninsula. But essentially, the Italian sparrow does maintain itself as a population that is recognizable, and its genome is stable as well. It's just that these independent hybridization events have sort of made a variety of genome possibilities. But that's what makes them so interesting, right? Because they all look like an Italian sparrow, but they've all got a slightly different genome. And that's what gives us a lot of power to try and understand the relationship between phenotype and genotype. There is one population of Italian sparrows that is really weird though, on an island called Tremiti. We think they're quite inbred, and that might explain it. And I don't have samples of them yet, but they have grey crowns. But their genomes are Italian sparrow, but they have a grey crown. But just to go back really quickly, you said, it's a species, and I don't disagree with you, but that's a massive philosophical question as well, right? So I'll throw it back to you and say, well, what is a species? Reproductive isolation is the general definition we use for a species, and it mostly works. But I think hybridization and this sort of leaky genes is probably way more common than we realise. And if you think broadly, it doesn't work for anything that's asexual, it doesn't work for bacteria. So I think humanity has been struggling with the what is a species question for many, many, many years. Yeah.
I know it's, for instance, in Icterine and Melodish warbler, is a lot of gene flow between those, especially in the hybridization zones.
Yeah, but that's what makes it so interesting, I think, right? There's still things which we can look at and say they look distinct. And for the most part, those boundaries work, but sometimes they don't. But they mostly work, so they remain distinct, at least in the period of time we're looking at them in.
It's our problem, eh? It's because we're twitchers and we want to know when it's a species.
Well, I don't know if it's just that problem. I think it might be an even bigger conceptual problem. We do like to categorise things, and maybe that's possible, maybe that's not. It's an epistemological problem.
All right, I have one question left over here.
When looking back at the Harlem bird, which you studied in hand, is there anything in its outward appearance that gives away that it's not an Italian sparrow? In other words, could we have seen it?
Okay, I actually think you guys are probably better at identifying many of the birds in the world than I am. I mean, I looked at the bird and I thought, yeah, it looks like the other Italian sparrows I've held. Size-wise, it didn't seem any different from a house sparrow, but then, you know, I also personally, I mean, the phenotype's really clear, but things like body size, I kind of need them side by side to be like, yes, this one's smaller. So, no, I was surprised by our results. I'm excited, but I was surprised.
Okay, thank you.
You're welcome.
Thanks. All right. Melissah, thank you so much for your stories. Then I would like to welcome Ruth on the stage. So, ladies and gentlemen, take a part.
Okay, hopefully you can hear me. Yeah? Thank you very much. Perfect. Okay, my name is Ruth and I work with Melissah at the Netherlands Institute of Ecology, also studying house sparrows. I'm very excited to be here with you today to tell you a little bit more about the work we do, mainly focusing, as Melissah said, on this house Bactrianus comparison, but I'll also be talking to you briefly at the end of the talk about the tree sparrow. As with Melissah, I'm obviously also speaking in English and I can talk quite quickly, so if anything is unclear, there'll be time for questions and clarification at the end of the talk. So let's get talking about some cool birds. So I know a lot of you here today are interested in the rare and the beautiful birds of the Netherlands and beyond. In fact, I think later on there's a talk about the rare birds that have been sighted in 2023. And I've been lucky enough on my travels to see plenty of rare and, in my opinion, pretty interesting birds. But when it comes to my research, these aren't the birds that I'm most interested in. I'm more interested in the everyday birds, I guess. The birds that when I'm doing my dishes on an evening, I can look out of my kitchen window and see, or those birds that I trip over in the city centre as I'm rushing through town to meet my friends for coffee on a weekend. I'm interested in the birds that have a close association with humans. And in particular, I'm interested in the subset of those that depend upon human resources to survive. So we can group these individuals or class these individuals that depend on human resources as being human commensal. And there's lots of examples of human commensals out there. Of course, for today, I've put some examples of human commensal birds on the slide, but there's also examples across a variety of systems, so we can think of mammalian examples like the black rat, the brown rat, and the house mouse, or other pests like bed bugs and fruit flies. And what makes these individuals different is that this intrinsic dependence on human resources. So if the people disappear, the human commensal population also disappears. So this is different to something like a great tit, for example, that might opportunistically use your bird feeder, but predominantly lives in the forest, and if you removed the bird feeder, the great tit population probably would sustain. And And there's two particular groups of human commensal that I'm interested in and we'll be talking about today. The focus of the presentation is of course going to be on the house sparrow, but the last couple of slides I will come back to the tree sparrow. So the house sparrow has had a long history of association with humans. In fact, we think house sparrows first associated with humans during the Neolithic. The Neolithic Revolution happened about 10,000 years ago and was a time of huge change within human societies as they transitioned from hunter-gatherer-based lifestyles to developing fixed settlements and establishing agricultural practices. And this, of course, brought about a huge change in the local environment for populations like the house sparrow that were able to interact and persist. So fixed settlements, for example, gave new nesting opportunities in the crevices of buildings, and there was a change in food resource abundance. So instead of the landscape being covered by natural grasses, suddenly with the rise of agriculture and domestication, we started to see an increase in the abundance of starch-rich cereal crops, things like corn and wheat. And these were different from the natural grasses in that they're a lot tougher and they have a much higher starch composition. So we think this transition likely led to dietary adaptation within the house sparrow. So the hypothesis that I'm working on for my PhD is that house sparrows have undergone some form of dietary adaptation to thrive as human commensals. And there's a lot of different ways that we can study the diet. First of all, we can see whether human commensal populations do eat more human foods. So basically, we can look back through their poop, because the easiest way to work out what goes into a bird is to look at what comes out the other end. And then if we do see a difference in diet, we can link this to a range of potential traits. We can look at the form of the bill, so I'm sure a lot of you are aware of the famous Darwin's finch example of different islands and different dietary resources being linked to differences in bill size and shape. And we can also link this to differences in bite force, so how hard the birds bite, which I'll come back to in a moment. We can also look at the guts of the birds and see whether the enzymes that are involved in digesting starch are in a greater prevalence in the human commensal population. We can also look at whether the birds have good gut bacteria for digesting this starch. And obviously, we also do a lot of genetics work, which I won't go into because Melissah's probably genetics you all out. So, in practice, how do we study dietary adaptation in the house sparrow? Well, as Melissah already told you, we go all the way to Kazakhstan. And you might be thinking, why on earth are you going all the way to Kazakhstan to study house sparrows? Because I've got about 100 in my back garden that you could do your work with. But it's actually because we wanted to try and get this elusive Bactrianus sparrow. Because to be able to understand human commensalism, we need a non-human associated comparison. So we went looking for this subspecies. It's ecologically very different to the house sparrow. It migrates from the Indian subcontinent up to these countries in the Middle East like Kazakhstan and Iran for the breeding season. And as you can see, the environments that we catch the Bactrianus sparrow are very different from the environments we catch the house sparrow. So we went to the Chokpak ornithological station in Kazakhstan that had these huge migratory traps, and then we could catch the Bactrianus sparrows as they were coming in to breed. As you can see, we were in the middle of nowhere, we were camping out with no electricity, no toilets, it was very glamorous. And we could look at the birds in this steppe environment and compare them to local town and city populations in catching from barns and places where you find people. And we collected, of course, a lot of data from this field season. It's not very often you get to go to Kazakhstan, so a lot of this data is still being processed. But today I wanted to talk to you a little bit about the bite force differences that we see between human commensal and non-commensal populations. So this is the special device that we use to measure bite force in a bird. It was developed by Anthony Harrell, and it's a pretty simple system of 2 metal plates that the birds bite down on, that's then linked to a force transducer, and we can measure the biting force in newtons. And we do this, as you can see in the picture here, at the tip of the beak, but also, yes, a little bit further back at the base of the beak. And with this, we can look at whether there are bite force differences between the human commensal house sparrow and the non-human associated Bactrianus sparrow. And when we look at the tip of the beak, we don't see a huge difference in bite force between the 2 groups. But when we look at our measurements at the side of the beak, we do see a more exciting difference. So the house sparrow has a significantly larger bite force than the non-human associated Bactrianus. And we would expect, if we were going to see a difference, that we would see it towards the base of the beak, because obviously these birds are seed eating, so the area of the beak that's responsible for crushing seeds is a lot further back in the bill. We might expect a difference in the tip with a raptor species, for example, that uses the force to tear meat foods. But why is this difference in the bite force at the base of the beak between the human commensal and the non-commensal so interesting? Well, this could represent a biomechanical adaptation to the difference in food processing requirements. So, as I said, the house sparrow is eating more human foods, so these tough cereal grains, which requires a bigger force to crack through than the soft and natural grass seeds that the non-commensal Bactrianus sparrow is eating. So it's quite a simple story of eating more human food means a reliance on tougher seeds, which means a bigger biting force. And this might be linked to differences in their jaw musculature. So it might be that the jaw muscles are of a larger mass in the house sparrow, which means that they're better able to eat these human foods. So that's the story in the house sparrow, but as I promised, I'm going to come back and just talk briefly about our upcoming research plans this year, shifting our focus from the house sparrow to the tree sparrow. Now, the tree sparrow is really interesting because it shows geographical variation in human association. So in East Asia, the tree sparrow is human commensal and essentially replaces the house sparrow being found in the towns and the cities. Whereas I'm sure a lot of you already know the tree sparrow in Europe is a lot harder to find. You will basically never see a tree sparrow in the cities and the towns in the same way as a house sparrow. And largely in the Netherlands and Europe generally, the tree sparrow is non-commensal. So we can perform a similar comparison that we've done between the house and the Bactrianus sparrow to look at whether there's similar adaptations to human resource use in the tree sparrow as in the house sparrow. And for this, as with Melissah's talk, I have a little question for you all. So we, as I've just mentioned, have acknowledged that there's not that many tree sparrow populations in the Netherlands. But we're in a room full of probably the best birders in the country. So if you know a tree sparrow population that would be good for us to study, please have a chat with us, tell us at the end of this talk, or send me an email if you prefer, and we would really like to hear from you. So that's the end of my talk for today. I'll end and say thank you very much.
All right, perfect. Thank you very much. Are there any questions around in the No questions so far. I was wondering, by the way... oh, I see a question. Sorry, I wasn't looking at the ball. There we go.
I wonder, can you use the whole genome information to calculate back when, for instance, house sparrows as a population exploded associated with the Neolithic transformation? I mean, if that is only 10,000 years ago, you would expect to be able to find a kind of genetic signature when you have so much genome-wide information, which from an evolutionary point of scale is rather recent. 10,000 years is really nothing, but it is a model which perhaps you could test if you could pinpoint that, because if it is a commensal, you would expect a population explosion in house sparrows.
Yep, so our group has published work on the past about this to link the divergence between the house and the Bactrian sparrow to the Neolithic biogenomics with a single origin hypothesis, so that there was a single origin of human commensalism within the Middle East, and then the sparrow population spread with agriculture. Now, with increasing genomic evidence, we think that hypothesis might be changing, so there might have been multiple independent origins of human commensalism, and then there's potentially different lineages of house sparrows that have independently come to human commensalism, and we can see that through the genomes.
Right. Any further questions over here? No? Okay, then I would like to thank you so much, ladies and gentlemen, Ruth Fawthrop,